Welcome to This Explainer. Today we're doing something a little bit different. We're taking a cinematic zoom inward. I mean, way past what you see when you look in the mirror, all the way down into the microscopic foundations of life itself. We're going to explore this bustling, totally invisible universe that makes you, me, and literally every living thing possible. Now, instead of just running through an agenda, I want you to think about this. How on earth did life actually emerge from boiling, chaotic water billions of years ago? And what exactly are these invisible, highly structured building blocks that put us together and keep us going today? It's this incredible mystery of survival, adaptation, and microscopic architecture against all odds. Okay, let's dive into this. Picture early Earth, about 3.5 billion years ago. Obviously not a very calm or welcoming place, right? A lot of researchers actually think life didn't kick off in some gentle, vast ocean, but in extremely volatile spots. Think of places like the hot springs of Puga Valley in Ladakh, India today. In these incredibly harsh waters, which are basically sitting right at the boiling point, you have organisms surviving just fine. These are heat-loving single-celled bacteria known as thermophiles. And honestly, it's just awe-inspiring. The fact that these tiny, solitary cells thrive where practically nothing else can gives us a huge clue about how life might have sparked into existence on a very turbulent young planet. But here's the catch. Life in a boiling, radiation-soaked environment absolutely needs protection to survive. So scientists studying those hot springs noticed something wild. Calcium carbonate was rapidly forming all around them. These mineral formations were basically nature's prototype for the very first protective membrane. They essentially shielded these fragile, early organic molecules from harmful radiation and deadly extremes. It was the crucial barrier that actually defines what a cell even is. So, if cells are the fundamental units of literally all life, why did it take humanity so long to figure out they were there? Well, the answer is kind of funny. Our own biology just kept us completely blind to them. Take the number 0.1. If you grab a piece of paper, draw two tiny dots, and hold it about 25 centimeters from your face, your eyes can only see them as two separate dots if they're at least 0.1 millimeters apart. Anything closer, it just blurs into a single point. And since cells are vastly smaller than 0.1 millimeters, they stayed completely hidden from the naked eye for almost all of human history. But that complete blindness to the microscopic world finally changed in 1665. Enter Robert Hooke. Using a light microscope he actually designed himself, which could magnify things maybe 200 to 300 times, Hooke decided to look at a pretty unimpressive everyday slice of cork. And what he saw literally revolutionized science forever. He found these tiny, empty, box-like compartments just stacked right up against each other. And because they reminded him of the sparse, tiny rooms that monks lived in, he called them cells. With that one incredible observation, the invisible building blocks of life finally had a name. Now, since Hooke's time, our tech for peering into this hidden world has just skyrocketed. I mean, compare the classic light microscope you probably remember from a school lab to the massive modern marvel we have today, the electron microscope. Instead of using visible light, this absolute powerhouse shoots a beam of electrons to produce highly magnified images. This technological leap lets us see the breathtaking, fine details of cell structures at the nanometer scale. That's 1/1000th of a meter. It's literally the difference between seeing the blurry outline of a house from a mile away and walking up to inspect the doorknobs. But hey, here's a brilliant little scientific hack for you. You don't always need a multi-million-dollar machine to measure a cell. With a basic microscope, a clear millimeter ruler, and a simple onion peel, you can easily estimate their size yourself. It's like a fun puzzle. You just measure your total field of view, count how many cells fit perfectly across that diameter, and do some simple division. So if your field is 5,000 micrometers wide and you can fit 25 cells across it, boom, each cell is about 200 micrometers wide. It's this elegant DIY math that lets anyone measure the microscopic universe. All right, let's zoom back in and focus on that crucial boundary we touched on earlier, the cell membrane. This barrier is unimaginably thin. We're talking just 7 to 10 nanometers thick. Yet this is the only thing keeping total chaos out of the cell. It defines the individuality of the cell and regulates absolutely everything that comes in or goes out. To understand how something so thin can be so incredibly effective, scientists use what's called the fluid mosaic model. This basically says the membrane isn't just a solid brick wall, but a flexible, fluid lipid bilayer filled with active, moving proteins. Now, what's really interesting about this slide is that this fluid mosaic is totally dynamic and restless. It's not static at all. It's made of two layers of fat molecules. You've got water-attracting heads facing the outside and water-repelling tails hiding safely on the inside.
Because of this unique chemical setup, the molecules aren't locked in place. They're constantly moving sideways, flipping and rotating, keeping the whole membrane fluid. Meanwhile, those embedded proteins, they're acting as selective gatekeepers, constantly communicating with the environment and deciding exactly what nutrients get in and what waste gets kicked out. They are arranged just like shifting tiles in a mosaic, constantly adapting to keep the cell alive.
To actually see this selectively permeable gateway in action, let's step away from the microscopic theory for a second and look at some macroscopic evidence. This is the famous potato experiment. We start out with two perfectly identical, freshly cut pieces of potato. They've been carefully weighed and measured right before we start, just to make sure they are exactly the same size. Next up, we take one potato piece and drop it into a beaker containing nothing but plain water. The other piece goes into a second beaker, but this one has a highly concentrated 20% salt or sugar solution. Then we simply walk away. We leave them completely undisturbed for about an hour. Now think about those tiny, fluid cell membranes inside the potato pieces. What do you think they're doing in response to these two drastically different environments? And this brilliantly illustrates the invisible power of the cell membrane. Just look at these dramatic physical changes. The potato piece that sat in plain water has visibly swelled up. It's plumped and way larger than before. But the potato we pulled from that harsh salt solution, it visibly shrank and completely lost its firm shape. Basically, billions of microscopic cell membranes were working overtime in there, causing the cells themselves to literally expand with water or entirely collapse. This physical transformation all comes down to osmotic pressure. So if a cell is in an isotonic environment, the concentration of solutes outside perfectly matches the inside. The cell's in perfect balance, so you see no change. But in a hypotonic environment, like our plain water beaker, water aggressively rushes into the cell, which swells it right up. On the flip side, a hypertonic solution, like our concentrated salt beaker, literally sucks the water out, causing the cell to shrink and collapse. That tiny membrane is constantly fighting to maintain balance against these massive external forces. But, not all cells react to these forces in the exact same way. Notice the contrast here. If you look at plant cells taken from an onion peel, they look like neat, rigid bricks, perfectly arranged in a sturdy wall. Human cheek cells, though, they are scattered, kind of irregular, and they completely lack any distinct uniform shape. So why does this drastic structural difference even exist? So the crucial point is that it all boils down to one extra layer of microscopic armor, the cell wall. Plant cells, fungi, and bacteria possess this rigid outer layer that sits just outside their fluid membrane. It acts like an exoskeleton. It gives them strict shape and vital protection, especially keeping the cell from bursting if it takes in too much water. This is exactly why plants have sturdy, crunchy structures, like tree trunks or celery, without having bones. Animal cells, like ours, entirely lack this wall. That's why we're squishy and flexible, but also incredibly vulnerable to shrinking rapidly if we're placed in a highly concentrated environment. We've journeyed from the boiling hot springs of early Earth all the way to the delicate, shifting mosaic of the modern cell membrane. We've seen how these microscopic entities shield themselves, how they communicate, and how they dramatically alter their shape just to survive their environment. But as we wrap up this explainer, I want to leave you with a profound question to ponder about these fundamental building blocks. Do these bustling microscopic cities, our cells, live forever? Or do they eventually die? And if they do die, what dictates their ultimate lifespan? The journey into the microscopic world is endless, and I hope this exploration leaves you eager to uncover more of its mysteries.